Then the steps of translation are three: initiation, elongation, termination. Initiation, the small and the large ribosomal subunits will bind together, moving along the messenger RNA, looking for the first AUG. Okay, and then the transfer RNA اللي هو specific for AUG having anticodon UAC will come and bind on methionine. بالelongation, this is the P site, this is the A site. تيجي transfer RNA للسكند كودون بتعبي ال A site. لما اثنيناتهم ال A وال P sites are occupied, I will have formation between them, formation of a peptide bond. بعدين حتمشي لنا the ribosome one codon. بس تمشي one codon ال A site حيصير عندي فراغ، واللي كان بال A site حيندفش لل P site لحتى أذكركم. اوكي، إذا بدها تمشي one codon بهيدا الشكل. So this is لهون one codon. اوكي، عندي اللي كانت هون بال A site حتصير بال P, and this is now the A site. A site is empty and ready to be filled by another transfer RNA. هيدا الشيء بكفي لأوصل ل one of the stop codons. I have 3 stop codons. واحد منهم حيكون موجود لحتى ينهي عملية translation. As you can see, the small and the large ribosomal subunits, they will dissociate, and I will have the release of the peptide and methionine will be liberated. Okay, نحن حضرنا one animation the last time. هلا حنحضر animation تاني. خليكن معي. اوكي، so يلا هيدي خاصة بال mechanism of the translation. نركز فيها منيح حتى نتذكر. You can see the video and hear the sound?
Yes.
Yes, teacher.
Okay.
Translation is the synthesis of a protein from an mRNA template. This process involves several key molecules, including mRNA, the small and large subunits of the ribosome, tRNA, and finally, the release factor. The process is broken into 3 stages: initiation, elongation, and termination. Let's see the process in action. Eukaryotic mRNA, the substrate for translation, has a unique 3' end called the poly A tail. mRNA also contains codons that will encode for specific amino acids. A methylated cap is found at the 5' end. Translation initiation begins when the small subunit of the ribosome attaches to the cap and moves to the translation initiation site. tRNA is another key molecule. It contains an anticodon that is complementary to the mRNA codon to which it binds. The first mRNA codon is typically AUG. Attached to the end of the tRNA is the corresponding amino acid. Methionine corresponds to the AUG codon. The large subunit of the ribosome now binds to create the peptidyl, or P site, and the amino acyl, or A site. The first tRNA occupies the P site. The second tRNA enters the A site and is complementary to the second mRNA codon. The methionine is then transferred to the A site amino acid, the first tRNA exits, the ribosome moves along the mRNA, and the next tRNA enters. These are the basic steps of elongation. As elongation continues, the growing peptide is continually transferred to the A site tRNA, the ribosome moves along the mRNA, and new tRNAs enter. When a stop codon is encountered in the A site, a release factor enters the A site and translation is terminated. When termination is reached, the ribosome dissociates and the newly formed protein is released.
Okay, now I will move to another. هيدي الانيميشن حتذكركم باتنين ميكانيزم مع بعض. انا قصد حطيتها هلا حتى تشوفوا ترانسكريبشن وترانسليشن مع بعض. يلا.
DNA is very long and cumbersome. The strands of DNA also code for many different proteins. To get the code for the desired protein from the nucleus to the ribosomes in the cytoplasm, a copy of the small necessary section of DNA will be made. The copying process is called transcription, and it occurs in the nucleus. It's the first step in protein synthesis, the process of making protein. The second step is called translation. A portion of the DNA is unzipped so that the mRNA can be made from the DNA like a template. That's transcription. Nucleotides of RNA match with one strand of DNA and make mRNA. RNA polymerase unzips the DNA and puts RNA nucleotides into the right place. C and G nucleotides match up, A and T nucleotides match up, but anytime the RNA places a complement to an A on the DNA, it places a U instead of a T in the RNA. Remember, RNA uses uracil instead of thymine. So, if the DNA strand looked like this, the complementary mRNA strand would be the complement, but each time there would normally be a thymine, there's a uracil in its place. The mRNA also doesn't copy the entire strand of DNA. It only copies the portion of DNA that codes for the protein it wants at the time. In fact, most of DNA doesn't code for anything. Only about 2% of it codes for protein. So the small section of coding DNA has a start and stop signal. After the mRNA is formed, it moves out of the nucleus through nuclear pore and goes into the cytoplasm. Remember, there are 3 different types of RNA. The purpose of the mRNA is to get the protein code out of the nucleus without pulling the DNA out of the nucleus. Then the mRNA will need to find a ribosome in the cytoplasm. Ribosomes can be found on the rough ER or floating in the cytosol. mRNA is read 3 bases at a time, and these 3 bases are called codons. Now, the ribosomes are made of protein and RNA, and the ribosomes are the protein-making machines that read the mRNA code and add the correct amino acid using tRNA. Remember, tRNA stands for transfer RNA, and that's because its purpose is to transfer the right amino acid to the ribosome to build the protein. The tRNA has an anticodon on one end, which will match a specific codon on the mRNA, and has a specific amino acid on the other end. Together, these 3 parts will make translation happen. Translation occurs in the cytoplasm of the cell wherever there's a ribosome. To get translation started, mRNA attaches to a ribosome, and a start codon must be read. It's usually AUG, and I remember that school starts in August, and that helps me remember AUG. The first amino acid is brought in by tRNA. The anticodon on tRNA matches up to the codon on mRNA. Then the next tRNA molecule moves in and matches up with the mRNA codon. This time, the amino acids form a peptide bond and link together. Then, the first tRNA can detach, and the mRNA shifts through like ticker tape, and the next tRNA molecule can come in. The protein grows until a stop codon is reached. Once the stop codon is read, the protein is formed and ready to finish folding to become functional. And that's the end of protein synthesis. Now, we can figure out the sequence of amino acids using the mRNA and amino acid chart like this one. Let's use an example. Starting with the first 3 letters, AUG, we can find the amino acids. The first letter is A, which narrows it down to this row. The second letter is U, which narrows it down to this column, and the third letter is G, which means that this is methionine, which is the starting amino acid. Next is CCC, which is the second row, second column, and second row on the right. But we don't even have to look at the third letter in this case, because every third letter will bring the same amino acid, proline. Next is GGC, fourth row, fourth column, second row on the right. That gives us glycine. And lastly is UAA, row 1, column 3, row 3, and that gives us stop. There's no amino acid here. It just means that the protein will detach, and it marks the end of protein synthesis. Thanks for watching this episode of Teacher's Pet. Don't forget to like and subscribe and follow me on Twitter at
Okay. So, as you can see, Ala rejoined. Ala Tattan. You are late, Miss. Hassan, you are late. Hawra Bassam, also late. Ala Hassoun, late. Yeah. اوكي انا بعدلي بس اخر فكره بالدرس بدي احكيها وبترجع ببعث لكم الفايل حتى تكتبوا the mechanism. I have something called polysome. It's polysome, or we can call it polyribosome. Okay, it's polysome or polyribosome. Poly means many. So poly means many. Many ribosomes. The definition of a polysome, it is having many ribosomes moving along the same messenger RNA. Thus, they are translating the same protein. Again, polysome is having many ribosomes moving along the same messenger RNA, thus having يعني بيطلع عندي كتير proteins. اوكي طيب انا بدي اسال هلا if I have 10 ribosomes moving along the same messenger RNA, knowing that each ribosome translates one protein. إذا أنا عندي 10 ribosomes, how many proteins will I obtain?
10 proteins.
10. طب are they of the same sequence identical ولا they are different?
What, Miss?
اوكي هعيد السؤال.
different.
هعيد السؤال. إذا أنا عندي هيدي messenger RNA اعتبروها هيدي صفحة ورقة. اوكي إذا بدي يترجمها 10 مترجمين قلتوا لي بحصل على 10 ترجمات 10 بروتينز. اوكي هال10 مترجمين اللي عم بيترجموا هن اكيد عم نحكي رايبوزومز وجواتهم الترانسفير ار ان اي عم بيكونوا. اوكي are these 10 proteins similar or different?
Different.
Similar.
عشان نفس الورقة.
صح برافو نفس الورقة نفس المعلومات. It's not different, it's identical. اوكي أنا عم بحصل إذا أنا بدي الإنسولين during transcription I told you many messenger RNA will be formed وهلأ عم بقلكم during translation many ribosomes will move along the same messenger RNA translating the same protein. So أنا بيطلع عندي البوليزوم or the polyribosome هني group of ribosomes moving along the same messenger RNA. They are translating the same protein. وأعطيت الexample if 10 of the ribosomes are moving along the same messenger RNA, then 10 proteins will be produced having the same amino acid sequence. They are identical. I need you to memorize this figure. Okay, polysome أعرف سميها وأكيد label أي شيء خاصه بالليبل كمان أنا مسؤولة عنه. Okay, عندي رايبوزومز. شوفوا كل رايبوزومز اللي بتكون مسبقة عن اللي قبلها عم بتكون عندها longer chain of peptides. اوكي، لأوصل لطرف المسنجر RNA أنا بحصل على complete peptide chain. Is it clear?
Yes.
اوكي، أنا هلأ رح أرجع لورا. في عندي آخر نوت. أنا عندي 3 steps of protein synthesis. I have 3 steps: initiation, elongation, termination. لازم تعرفوا إنه ال translation process أو ال protein synthesis it requires energy. هي بحاجة للenergy. Which of the 3 steps is it? Initiation, is it elongation, or termination? Which of the 3 steps you think it requires the highest amount of energy?
termination.
فكروا فيها، أي فيها أكتر شي شغل؟ أي فيها أكتر شي شغل؟
elongation.
صح. initiation والtermination أقل شي فيهم شغل. لأكتر شي في شغل هو elongation. So elongation requires the highest amount of energy. elongation. So أول part من النوت elongation requires the highest amount of energy. هلا تاني جزء من النوت this is the elongation. سمولي الاشياء اللي بالelongation يلي هي بتخليها most requiring energy step of the protein synthesis. في شغلتين، في سببين. طلعوا بالرسمة فتشوا على 2 reasons, 2 factors making the elongation needing this amount of energy. What do you think? يلا I need answers. يلا مس قولوا اللي ببالكم ما بيأثر.
complex.
ايه complex شو في تحتها؟ complex شو؟
First peptide bond.
اوكي، فإذا البيبتايد بوند أول سبب هي البيبتايد بوند. تتخلي الإلونجيشن هي needing this amount of energy هي البيبتايد بوند. So البيبتايد بوند تحتاج كتير energy. وعم نحكي نحن كل ما يكون عندي 2 amino acids حد بعض حيصير عندي formation of peptide bond. This is the first factor making elongation needing energy. شو تاني factor برأيكم كمان؟ What do you think the second factor is? بهالرسمة
لما عم يصير عنا realize breaking bond.
no عم بتصير عنا شو؟
of ribosome.
عم تمشي الرايبوزوم. the translocation of the ribosome. أهم خطوتين هن بالإلونجيشن. بيتعبوا السايتس اثنيناتهم، بس يتعبوا بده يصير في بيبتايد بوند وتمشي الرايبوزوم. صح؟ فإذا البيبتايد بوند والtranslocation of ribosomes which means the movement of the ribosome, these 2 factors makes the elongation the most requiring energy step in the protein synthesis. So the peptide bond and the translocation of the ribosomes during the elongation step of protein synthesis require the most amount of energy. Okay? Okay, miss?
Okay.
Okay, teacher.
اوكي، خليكم معي